Hi, thanks everyone. Thank you for that introduction as well, which to what is my ultimate mission, to bring you all to Canada. I'm here to recruit you, where we can do a little bit, we have a little bit easier access to early phases of research in cannabis-based medicine. So I'm here to represent my organization, Santé Cannabis, our small team here that contributed to this work, but our larger team back home in Montreal, Quebec. So just some disclosures. I'm going to present about what is an investigational drug, which is owned by sponsor Tetra Biopharma, and some work that was done at a large CRO, North American CRO. So we've talked a bit about Canada. We've got some excitement happening, both with the introduction of the Cannabis Act last week, which legalized cannabis for adult use. But we have a proliferation of licensed producers and manufacturers of products in Canada that are regulated federally. And we have access to the machine of regulatory investigational processes that allow the development of cannabis-based drugs governed by the same division that looks at the development of other pharmaceutical medications. We also have, with the Cannabis Act, a lot of opportunity for smaller cultivators, processors, manufacturers to get into the game, as there's now an inclusive framework for different sizes of production and facilities. So in the Cannabis Act as well, we saw a streamlining of access to cannabis products for research purposes, and this should make working in this field much easier moving forward. So we don't have really a huge revolution happening, though. We've had a lot of cannabis dispensaries, so a lot of the hype around, how much of a change this is on the ground level, it isn't really founded, because we've had gray area dispensaries, hundreds of them operating. It's more of a shift for those of us who've been working in this field for a long time to suddenly see these dispensaries closing out sales in preparation for what are government suppliers to take over. So this is the Ontario retailer here in our largest province, and quotes on Twitter just astounded by the interest in their products, which we all know very well. So cannabis-based medicine in Canada, obviously we have a lot of the same opportunities and challenges that you see here, but importantly, if we look at a lot of the challenges over here, we decide that we can look at these access to information, research, education, training, stigmatization, and take the focus on the opportunity of developing research products and investigational drugs to address this long list of challenges that we have. So that brings us to our mission at Santé Cannabis, where we exist in Quebec. Santé, very important for you to know, means health in French. I've had to translate to quite a few Americans this week. And we facilitate a series of clinics across the province where we're supporting patient access to physicians and our nursing team for support, education, and treatment plans, but we're also collecting a huge battery of different research outcome measures. So to do that, it does involve quite a few different people, but this is a glimpse at our clinic and research model. So we initiate with a referral from a physician treating the patient in primary care, and then run that through our system to initiate the patient into our research database. The patient will be seen in person at the clinic by a nurse who will do a complete assessment and be able to collect a huge list of baseline data. The nurse works together with the physician to implement the treatment plan for that patient. And then critically, we have our core of our team, the nurse educators who provide education, follow-up, support, modification of dosages and product recommendations ongoing. And this is all adhering to a physician standardized treatment plan. So what that means is then we're able to take that experiential data or observational data which we're collecting from the patient, and take that over to pharma sponsors, small biotech companies, experts in drug development who are looking for keys to their development, and start to initiate a process where we can develop cannabis-based drugs in parallel with our work at the clinic. So as we know, there's a very complexity of treatment options. We have more than 100 producers in Canada, each producing between 10 and 50 different products. So this translates into a huge number of products. We start to then categorize the products that are available into different formulations of THC, CBD, and try to collect as much data as possible about other cannabinoids and terpenes. And then we make decisions around route of administration selection, primarily between inhalation and oral, which is what we have available in Canada. Then if we dig into the patient use patterns at the clinic, we still see that we are in a transitionary phase. So in methods of administration, while oil or oral administration has been growing in popularity and is now our most common method of use, we see still a lot of inhalation, which has been persistent over time. So more than 50% of patients are using inhalation in some form, whether incorporated with oil or on its own. And in that category of inhalation, we still see a very persistent population that is insistent on smoking as well. So vaporization has been growing, but even patients who have a vaporizer often report that smoking can be more effective for them and are often using the two in tandem or in a kind of mixed model of inhalation. So this drives this question, what is going on with this smoking and why patients are still reporting such a difference between vaporization and smoking administrations? So the big question of inhalation is then, is it necessary? We have all of these other methods that are developing, especially here in California and other US states, where you have topical creams, sublingual sprays, patches, things like that. But we still see this persistent desire for inhalation, and especially if we think about specific symptom categories such as breakthrough pain, nausea, vomiting, appetite stimulation, and the potential for substitution as well of problematic substances of abuse. This kind of rapid onset of inhalation is thought to be very critical. So if we think about the ongoing development of inhalation as a method of administration, it's our goal to really go back and look at the root of the issue. So inhalation methods, though we have a lot of information that has been published about their in vitro results, such as using an artificial lung, analyzing components in the smoke or the vapor, we don't have enough information about the in-human response to these different methods of administration. So this brought us to work with the sponsor I'm highlighting here. This is the product that was developed for analysis and comparison between smoked administration and vaporized. It's named as the investigational drug PPP-001, active pharmaceutical ingredient both THC and CBD in a specific ratio. So this product is standardized as a blend of 3 different strains of dried cannabis at a dosage of 280 milligrams of whole plant material, and standardized to have a dosage of 25 milligrams THC and 5.5 milligrams of CBD per pellet. Then in the placebo, we have 0 THC and a small fraction of CBD. So the objective of this first study, which I'll present today, is to evaluate the safety and tolerability of this fixed dose in an inhaled or smoked THC CBD administration over 1 day and then over 7 consecutive days. The methods involve a kind of clear standard process for a phase 1, double-blind, placebo-controlled. So we'll be looking at single and multiple daily doses in both of those cohorts. So importantly, this was a very popular study in recruitment because subjects were confined for monitoring during the entire duration of the period and for 36 hours pre and post. So it involved a lot of video games and being paid quite well for that time. This was approved according to standard procedures, and we're looking really primarily at safety outcomes. So vital signs, ECG, was done at various time points, physical exam, and a battery of clinical laboratory tests to look at different safety outcomes, including a standard process for cognition. The device that was used is an inhalation device approved actually in Canada as a Class 2 medical device. It's a titanium pipe which can be used to smoke cannabis. And this was a standardized controlled inhalation to try to get as close as possible to a standardized dose. So it was very carefully instructed to inhale for 3 seconds, hold for 3 seconds, exhale, and then repeat the process for as many inhalations as required to complete the dose. So in the first cohort, these patients were looked at, were divided into groups of 8 subjects. Dosage was first just once daily for 1 day, and then moving to twice and 3 times a day with the same product. Then that safety data would be analyzed before proceeding to the second cohort, which would essentially progress through that same process over a 7-day period. So importantly, in the demographics, we had a pretty good range of age from 25 all the way up to 60 years old, primarily 60% male, primarily Caucasian, average BMI, and pretty standard other measures of health. So when we look at the pharmacokinetics, this is where things start to get really interesting when we look at smoked administration. We had THC and CBD detectable in the first blood draw, 1 minute after inhalation. And for most subjects, this remained for about 3 hours, though many of them had almost complete elimination after 1 hour post-administration. The maximum concentration, or Cmax, was very variable, even with the very standardized method of inhalation that we employed, with a maximum concentration of 30 all the way to 180 nanograms per ml. Now, 180 is something that's very rarely observed. I think there's one report of such concentration in a clinical study. Usually we see 50 to 60 nanogram per ml as a Cmax in vaporization studies. So the maximum time on average was 5 minutes. So that's the peak plasma level occurring at 5 minutes post-administration. And in this cohort, we didn't see any accumulation effect at all. So before the next dose was administered, there was complete elimination of THC, CBD, and the metabolite alpha-11 hydroxy. We also didn't see any steady state. So in pharmacodynamics, we can see that there's an impaired cognition actually after the first administration, but that this actually resolved as we were administering further. So we saw some development of tolerance, though in this small population it can be difficult to make large estimates of that effect. We also saw some limitations that we weren't looking for functional impairment, but just a standard impairment, small sample size, and I'm not sure yet about clinical significance until we move to our next round of studies. So something that's very interesting in the data is if we look exactly at these different subjects. So this safety effect, this safety report actually stopped the study periodically, a severe adverse reaction and 2 moderate ones, which involved the subject fainting. Now, this was a 52-year-old Caucasian female. She'd been exposed to cannabis in the past, so was used to the effects. And you'd expect her to have a very high concentration in her blood, but actually she was below average with just a 52 nanogram per ml. So this is really showing the difference between subjects and how this response can be also very psychosomatic. There were no clinical reasons why this subject should have fainted. And when we think about the dosage of just 25 milligrams of THC, it's quite shocking that someone could have this effect from that dosage, considering the doses that are sold and used in dispensaries and other clinical settings. So in comparison, this subject, 25 years old, Asian female, with a pretty low birth rate and low resting blood pressure, had very mild side effects, but had the very highest concentration in her blood. So her tolerance to the 180 nanogram per ml concentration was quite surprising. Same cohort, same dose, but a very inter-individual response. So importantly, and as many of you know, working in cannabis, the start low, go slow really holds true through clinical setting and through double-blind trials, where we saw that a titration strategy was necessary to progress to a 7-day administration after subject number 3 had such a response. So we introduced a slower titration to move subjects towards the full dose as they moved towards the 7-day administration. So they started with just 1 inhalation and moved up to completing the entire pellet. So this was done to satisfy Health Canada to progress to the 7-day administration and showed a significant improvement in the AE, the adverse event profile. So overall, you see here that these are normalized to the number of dosages that were administered. So actually, when that titration was administered, the start low, go slow process, we see that that cohort actually had lower instance of adverse events than the placebo did in the prior cohort. So we really see a significance there. Otherwise, the most common events are somnolence, dizziness, and headache, as well as some euphoria. So overall, this was a very successful 7-day administration. So importantly now for safety, really this big question is, can this drug be moved into phase 2 and 3? That was clear from the results. There was nothing clinically significant in cardiovascular, no drug-related changes in serum, hematology, or urine. And everything was primarily mild that was reported, 82 and 94% in the 2 cohorts. And we had no serious adverse events at all, nothing life-threatening or requiring hospitalization. So this product has moved into now a couple of different trials. And the first one, a phase 3 trial, which is really a compassionate use focus. We are recruiting for this right now in Quebec and are looking for other sites. So feel free to reach out to us.
3 minutes.
And finally, this was found to be generally safe and well tolerated. The protocol is to be repeated with a vaporizer next month to compare the different results and to see the differences in pharmacodynamics, which is very interesting. It's interesting as well to look at now why this concentration may exceed that what we've seen in vaporizer studies. So once we verify that, it will give us a better idea of in which populations smoked cannabis may be preferential over vaporized, and explain a little bit of why we see the differences in the patient reporting. And then finally, that this maximum time of very rapid delivery could be the most effective way to treat acute symptoms. Finally, a limitation that is worth a really heavy investigation in tech is that this variability in inhalation is going to be a huge challenge and will require some very innovative drug delivery systems. So here's the pitch. If you're interested to study this drug, please reach out to our company or to Tetra Biopharma, and it will be available as well internationally. And for any contacts, thank you very much.
Hi, that was a great presentation, Dr. Caroline McCallum from Vancouver, BC. I'm just wondering if you can elaborate a little bit more on the neurocognitive impairment, if there was a standardized questionnaire that was given to patients, or if it was a functional examination, or how you guys classified that.
There was a series of tests that were performed for both short-term memory, for small motor skills, as well as the kind of standard mini cognitive effects for cognition. But unfortunately, we didn't study functional impairment, which is something that we'll look at in future studies, as we would be concerned about certain populations, oncology populations, geriatric populations, to use this product without that assessment.
Great, thanks so much.